Hello everyone, thank you for watching our talk about siPOOLs and how you can improve your RNAi screens with the power of pooling. I'm Anna Lisner, the Head of Sales and Marketing at siTOOLs Biotech, and will give you the talk today. So let's jump right into the topic of RNAi. At first, I wanted to remind you of the benefits of RNAi. RNAi is widely used as a gene silencing tool to determine gene function across various biological contexts and systems, as it is fast, drug-like, and simple to apply. With fast, I mean that just within 24 hours, RNA loss is seen, and after a few days, a protein loss is detectable. And what I mean by RNAi is drug-like is that RNAi is dose-dependent and reversible. And RNAi can be used directly onto cells. It results in efficient uptake, therefore avoiding any clonal selection or cellular engineering, which brings me to the next benefit of RNAi. RNAi avoids genetic compensation that can occur under prolonged loss-of-function experiments. Last but not least, RNAi is broadly applicable. It can be used in cell lines in primary cells or in vivo systems with appropriate delivery vehicles. Naturally, no experiments come with only benefits. The drawback of RNAi is its off-target effect. Artificial siRNAs induce changes in expression patterns of genes that were not supposed to be targeted by that siRNA. This phenomenon especially occurs when using high concentrations of single siRNAs. Here in this microarray analysis, you can see how a single siRNA against STAT3 can result in numerous off-targets. Even a 1 nanomolar concentration of the single siRNA already leads to a difference of expression levels of other genes. And as you increase the concentration from 1 to 10 or even 25 nanomolar, you see that the effect gets even more drastic. More and more genes will get affected by this single siRNA, even though this siRNA was designed for just one gene. And as you might be wondering how the off-target effect occurs, this is the mechanism behind it. As you all know, endogenous siRNAs typically need the full complementarity for RNA target recognition and subsequent direct cleavage. This mechanism is wanted by all RNAi screeners. However, when increasing the concentration, artificial siRNAs can also be bound by organelle proteins, usually binding microRNAs. MicroRNAs are other endogenous non-coding RNAs like siRNAs. And the microRNA binding organelle proteins only need the 6 nucleotide long seed sequence of the microRNA for target recognition, as you see here in the below graph. To induce translational repress or target degradation, the seed sequence only needs to have near-perfect complementarity to the target, so wobble-based pairing is allowed. Therefore, the window for potential targets is opened by only having the seed sequence as necessary target recognition sequence. And here I wanted to explain to you how we conquer the off-target effect. And as I said before, single siRNAs lead to off-target effects with low or variable efficiency, especially when using high concentrations. This is demonstrated here with the green bar representing the target that is supposed to be targeted by the siRNA, and the ones above in the other colored bars, these are the off-targets. And as you see, single siRNAs also lead to knockdown of other genes, as we've seen before. How we as siTOOLs Biotech conquer the off-targeting effect is we have a high complex pool of siRNAs in one product, the siPOOL. Each siPOOL consists of 30 siRNAs, each having a different seed sequence and targeting the entire target transcript. Therefore, targeting is more specific, and due to low concentration of each of these 30 siRNAs, we dilute off or away the off-target effect. So the key is to reduce the concentration of each siRNA inside the siPOOLs to enable specific targeting and increase efficiency and reproducibility. Therefore, you save money and time when using siPOOLs for siRNA-mediated gene silencing, instead of using single siRNA or low complexity pools of siRNAs. And this pooling approach we also use for our other tools, the RA pools, which are used for affinity purification, and the RABO pools which are used for ribosomal RNA depletion in any species. And you can check out the RAPOOLs and the RiboPools in our other videos here on our YouTube channel. Here on this slide, I wanted to show you how well the siPOOL actually reduces the off-target effect with an example. So here on the left-hand side, you can see that a single siRNA leads to an off-target effect. The knockdown of the target gene that is represented here in green is sufficient. However, the red dark dots represent a gene that was not supposed to be targeted, but the expression levels were changed upon knockdown. And when using that same single siRNA that was used during the left screen in a siPOOL with 29 other siRNAs at the same concentration, you can see that the target gene was still knocked down. However, only a few other genes were affected. So this is a great example of how the siPOOL reduces the off-target effect to a minimum. And I want to give you one more example, and this is actually customer data from the DKFZ in Germany. The scientist Mona Malz wanted to knock down one paralog of a gene while not affecting the expression level of the other paralog. Therefore, we designed two siPOOLs, one targeting SMARCA2 and the other siPOOL targeting SMARCA4. The analysis was performed using RT-PCR, and on the y-axis you see the relative RNA levels. And as you see for both SMARCA2 knockdown and SMARCA4 knockdown, the siPOOLs were able to reduce RNA levels of the targeted genes at very low concentrations. For example, SMARCA2 siPOOL was efficient at a concentration of 0.02 nanomole. In summary, a concentration of 0.3 nanomole, which is really low, the siPOOLs produced a 95% knockdown for SMARCA2 and an 87% knockdown for SMARCA4, without any cross-reactivity between the paralog-specific siPOOLs. So in summary, siPOOLs are specific enough to efficiently and separately knock down paralogs. As you might be wondering, how can I validate my siPOOLs? This can be done with a siPOOL-resistant rescue construct. siPOOL-resistant rescue constructs are designed by wobbling single bases at siRNA recognition sites, which is shown here in orange, until sufficient mismatch is reached to confer siPOOLs resistance. When expressed with a DNA vector, the codon-optimized sequence will then rescue the loss-of-function phenotype induced by the co-administered siPOOL. In summary, a single siRNA result in variable results and therefore hinder data interpretation. And with our siPOOLs, scientists can save money and time as siPOOLs effectively counter the siRNA off-target effect and improve reliability of results. Moreover, we offer customer support whenever you face technical issues. Also, we are very transparent when it comes to siPOOL design. Thank you very much for your attention. In case you have any questions, we are happy to help. Just send us a mail at info@sitools.de or ask your local distributor. In case you need any more information about siPOOLs, we're happy to help as well. And you're not still convinced about siPOOLs? Well, scientists at Johnson & Johnson, Roche, Bayer, or Galapagos trust our siPOOLs, so why shouldn't you?